Hello everyone and welcome back to PhytoPathoVerse. I'm Malini, molecular plant pathologist with a motto to make plant pathology easy. Before we jump into chapter 2, let's quickly recap what we have covered in chapter 1 of GN Agrios. That is the history and development of plant pathology. Here we have seen how plant diseases were observed, how they were misunderstood, and how scientific breakthroughs shaped their field. Okay. As promised, I will be uploading separate videos on the major historical events. Also, I hope you have checked the memory-based takeaway questions I shared from chapter 1. Again, before moving into chapter 2, let's quickly revisit the questions I had asked you to search from chapter 1 of history section. Number 1, that is, which pathogen is responsible for causing ergotism or holy fire disease? That is Claviceps purpurea. Next, who is known as the father of virology? Beijerinck. Third, who is regarded as the mother of virology? Helen Purdy Beale. Last but not the least, who is considered as the father of molecular plant pathology? That is Kin. N.T. Kin. Now coming to chapter 2, effects of pathogens and diseases on plant physiology. In the last session we have studied is that physiology plays an important role whether a plant is healthy or diseased. How come? When the physiological function of a plant is carried out to the best of its genetic potential, then it is called healthy, right? When any of its activities are disrupted, altered, or inhibited, then a plant will be called as diseased. Simple. So today we will be learning about how pathogen affects the various physiological functions of a plant. Okay, let's get started. To start with, we have the introduction. Plant disease is the lasting departure from normal physiological functioning of plant caused by pathogen interfering with the metabolism of its host for the purposes of nutrition acquisition, reproduction, and survival. Okay. The effects of disease on the plant host manifest as symptoms that can vary in severity depending on the susceptibility of the host and the life strategy and aggressiveness of the pathogen. Here, susceptibility means what? Susceptibility means, in normal sense, we can tell vulnerable. Okay. Aggressiveness is what? Aggressiveness, normally we can tell like it is a quantitative measure, but we can term as severity of a disease. It is measured how aggressive that pathogen is. So host should be susceptible and pathogen should be um, aggressive. Not we cannot say aggressiveness because it is just a quantitative measure to quantitative unit to measure how severe a disease is. So okay, by that a disease is caused. Now pathogens differ with respect to species of plant that they attack, the organs and tissues that they can infect, and the growth stage of the plant they can colonize successfully. Now some pathogens are restricted to a single plant species as their host, others to one of the one genus of plants, and many have a wide range of hosts belonging to many families of higher plants. Okay, here we can see some plants, they have single plant species or as their host, means they are, we can say they are host specific. Okay, now there are few which have wide range of host. Okay, they are non-host specific. We will give one example of each. Okay, host specific, we have heard the term Puccinia graminis triticii. It is the black rust of wheat and it affects only and only on wheat. Because of this is because the we know the scientific name of wheat is Triticum mastifum, so it is specific to wheat. Non-host specific, there are many examples of non-host specific, but we will be studying the most highest means largest host range. One example of such kind, that is Sclerotium rawlsii. It has the highest, the largest host range, over 500 plant species across 100 families. 500 plant species as its host. It normally causes stem rot disease. Okay, so moving on, we have three terms here: parasitism, pathogenicity, and aggressiveness. Let's see what it's saying. A plant parasite is an organism that becomes intimately associated with a plant and multiplies or grows at the expense of the plant. Parasite means we know which organism which is parasitic to other organism, means they get benefited from the second plant. Okay, so they can't multiply there or grow, whatever they can do. A parasite secures nutrients and water from the host by modification and exploitation of host physiology, metabolism for the benefit of the parasite. I hope everybody is familiar with the term parasite. We have heard of it many a times. Okay. Now, based on the nutrient requirement and lifestyle, plant pathogens are traditionally classified as biotrophic, hemibiotrophic, and necrotrophic. Now, what is biotroph, biotroph, and necrotroph? Let's see. Okay. Biotrophs can only complete their life cycle on their host, diverting nutrients from a living host to support their parasitic growth and reproduction. Okay. Most biotrophs produce specialized infection structures such as Hosteria to obtain nutrition and suppress host immune responses. Biotroph means, they need living organism for their survival, and sometimes they um, produce specialized structures called Hosteria. We'll come to this point later. After that, we have necrotrophs. Necrotrophs can survive and grow on dead matter, such as artificial laboratory media or crop debris, but in the presence of a living host adopt a parasitic lifestyle and can be pathogenic to a few or numerous host species. Necrotrophs obtain their nutrition by killing or digesting host sacrificed cells, actively destroying plant biomass. Okay, that's it. So here, simply we can tell biotrophs need living thing for its survival, whereas necrotrophs, they survives on dead matter. Okay. So normally the pathogen that can be cultured in laboratory are mostly necrotrophs, because in lab we don't have a living thing. If we are trying to maintain a living maintain a biotrophic or obligate parasites, okay, so we need to maintain it in a living plant. Next comes the term hemibiotrophs. So the term hemibiotrophs describes a number of organisms that may have narrow range of hosts, keep alive during the early stage of invasion. However, once established on their host, this pathogen switch to necrotrophy, which means they initially they live as biotroph. Once they are established in a living structure, they kill it and they convert themselves to necrotrophs. That is a selfish kind of behavior, but that's okay. What we can do about that? The next term that we see here is pathogenicity. So pathogenicity is the ability of the parasite to interfere with one or more of the essential functions of the plant, thereby causing disease. Pathogenicity is a categorical term that describes whether a pathogen can cause disease on a host to any level. Simply, pathogenicity is the ability of a ability of pathogen to cause disease. Okay, and this is um, a process of disease. That okay, the process that I'm talking about. One more term is there, ki pathogenesis. That is the method or the mechanism or the process by which pathogen cause disease. But the ability of a pathogen to cause disease, this is pathogenicity. Okay. Now next one more term is left, that is aggressiveness. Yeah, here it is. The degree of damage measured as severity. I have told you earlier, severity is described as aggressiveness of the pathogen. If a pathogen causes severe disease in a rapid manner on many plant cultivars, the pathogen is described as aggressive. In many host-specific interactions, different strains of a pathogen can only cause disease on some cultivars of a plant and not others. Okay. Now, when there is a disease, the host pathogen interaction is compatible and the pathogen is described as virulent. This term will come frequently, okay? Susceptible, host, virulent pathogen, and favorable environment. Okay, these three are necessary for a disease to occur in a plant. So yes, when there is a disease in host pathogen interaction, it is called compatible and the pathogen is described as virulent. If the host is resistant to its strain of the pathogen, the host pathogen interaction is called incompatible. Understood? If disease is causing, then they are called compatible. Okay, if there is no disease, then the relationship it will be called as incompatible. Understood? This might come in objective questions, so remember this term. When disease occurs, it's compatible. When there is no disease, it is called incompatible. Okay. 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 And and the pathogen will be called as avirulent. When disease is caused, the pathogen is called virulent. When there is no disease, the pathogen will be called as avirulent. Now, the term avirulence and virulence are also categorical terms under the quantitative ones. Okay. Now, we will start with the amount of disease. A plant becomes diseased or functions abnormally in most cases when it is attacked by a pathogen, or when it is affected by an abiotic agent. Abiotic agent means what? Non-living agent, which is drought, flooding, heat, cold, herbicides, etc. In the latter case, the physiological damage is not caused by an infectious organism, and it is often referred to as disorder rather than a disease. Okay. Now, from starting, we are talking about pathogen, and disease. So yes, when there is an involvement of pathogen, then the symptoms that we will observe, that will be termed as disease. Okay. So disease is caused by any of the pathogenic agents, that can be fungi, viruses, bacteria, nematodes, protozoa, molecules, and so on. Now, but when there will be the involvement of abiotic agents, that is non-living agents like temperature, water, for temperature we have drought, for water we have flooding, heat, cold, herbicides, all the non-living parameters where they are involved, we will call them disorder but not disease. Okay. However, in the first case, for a plant disease to occur, at least two components, a plant host and a virulent and aggressive pathogen, must come in contact and must interact. Yes, if at the time of contact of a pathogen with a host and for some time afterward, conditions are too cold, too hot, too dry, or otherwise unfavorable, the pathogen may be unable to infect the plant, or the plant may be able to resist or escape the attack. And therefore, despite the two being in contact, no disease develops. Okay? Okay. Now, what they are saying is that from in the last thing, we are only talking about this pathogen, this, where is it? Okay, I'll just write it down. From the last point, we are only talking about pathogen and host, right? Pathogen and host. When there is a favorable, no, when there is a susceptible host and virulent pathogen, disease will occur. But what will happen if the temperature is high or low? It is not favoring the pathogen to survive. Like, there are so many pathogens which cannot survive in high temperature, okay, or in very low temperature. If the humidity is too high or the humidity is low, there is severe rainfall, or it can be anything, like, see, too cold, too hot, too dry, or any other uh, condition may apply. So in time of unfavorable condition, pathogen may be unable to infect the plant. So only these two parameters is not um, essential for a pathogen to cause disease. For that, what we will require? We will require a favorable environment. Here only I have told you susceptible host, virulent pathogen, and favorable environment. These three are required for pathogen to cause disease. 1, 2 is there, 3 is not there. 1, 3 is there, virulent pathogen is not there. Favorable environment, susceptible host is there. Again, means any of the three. If one is not in coincide with the other two, disease will not occur. Okay? Okay. Therefore, The environmental condition can alter the interaction in favor of the pathogen or the host. The three disease components have often been visualized as simple triangle, generally referred to as disease triangle. This is called disease triangle. Each side of the triangle represents one of the three components, and a fourth component can be added to represent the time. Fourth component can be added, that is time, over which the interaction occurs. The length of each side is proportional. Try to remember this. The length of each side is proportional to the sum of the characteristics of each component that favor the disease. Okay. For example, if the plants are resistant at the wrong age or widely spaced the host site, the amount of disease would be small or zero. Whereas if the plants are susceptible at a susceptible stage of growth or planted densely, the host site would be long and the potential amount of disease would be great. Understood what they are saying? Means this is the amount of disease they are saying. It depends on how much virulent a pathogen is, how much susceptible. For example, if a pathogen is not this much susceptible, they are this susceptible. For example, this much, not that much. Virulent. A host is this much, not excluding this part. We are excluding this part. And favorable condition is this much. So this see the area is less, right? So the amount of disease will be less. But if we compare these points, the amount of disease will be more. That's what they are saying. If the three components of the disease triangle could be quantified, the area of the triangle would represent the amount of disease in a plant or in a plant population. Therefore, when there is a sufficiently long period of favorable conditions for an aggressive pathogen to attack and infect a susceptible host population over a large geographical area, the amount of disease is so great that disease epidemic develops. Again, today we have used this term epidemic. In the last session, I have told you there are various terms called endemic, epidemic, pandemic, and sporadic. Knowing these terms are very important. Okay. Okay. Let's moving on to the disease cycle. In every infectious disease, a series of distinct events occurs in succession and leads to disease development and perpetuation of the disease and the pathogen. The chain of events is called disease cycle. So in the disease cycle, there are a series of events. The first will be dispersal and inoculation. Second, penetration and establishment. Third, colonization. Fourth, pathogen growth and reproduction. And the fifth is survival. Cool. So first we will start with this thing. Type of inoculum, source of inoculum, and survival. What is there? The pathogen lands on the plant and or is otherwise brought into contact with, it is called inoculum. Okay, so the plural is called inocula. The inoculum refers, here comes the definition, the inoculum refers to any part of the pathogen that can initiate infection. What it can be? In fungi, the inoculum may be spores, sclerotia, or fragments of mycelium. In bacteria, molecules, protozoa, viruses, and viroids, the inoculum is always the whole individual. In nematodes, the inoculum may be adult nematodes, nematode juveniles, or eggs. In plant parasitic higher uh, plants, the inoculum may be plant fragments or seeds. Okay. So, one unit of the inoculum of any pathogen is called propagule. An inoculum that survives dominant in the off-season and causes the first infection of in the spring or autumn, it's called primary inoculum. And the infection it causes are called primary infections. Pathogens that complete one disease cycle during their host growth and development are called, any guesses? It's very simple. One disease cycle. So, in one season, what it will be called? It's called monocyclic. The cyclic, it's called monocyclic. The pathogen that reproduce rapidly and complete their disease cycle multiple times during their host lifespan are called polycyclic. Also, in polycyclic diseases, secondary inoculum produced from primary infections that causes secondary infections, repeating the disease cycle on the same host. Now, there is some monocyclic pathogens that take longer than 1 year to complete their disease cycle and several years to build up primary inoculum. So, what they will be called as? They have different name, which is called polyatic. Okay. Now we will see some of the examples of monocyclic, polycyclic, and polyatic diseases. So monocyclic diseases, they are called most of the soil-borne diseases are monocyclic. Stem and root diseases are often called monocyclic. Okay, any soil-borne diseases you will heard of, all will come under monocyclic disease. While mildew, blights, all are polycyclic diseases. Many diseases of trees. Whenever we will talk about trees, okay, like vascular wills, they will be called as, they will come under polyatic diseases, which means they are basically monocyclic, but they require more than 1 year to complete their life cycle. So learn the difference, okay, monocyclic, polycyclic, and polyatic diseases. Okay. So, okay, we can move on to dispersal and inoculation. Monocyclic pathogens are often dispersed by air or rainfall. In contrast, polycyclic pathogens may rely predominantly on airborne or insect vector dissemination, causing severe epidemics in crops. Airborne inocula usually settle onto the plant surface by gravity, but may also be washed out by rain. Okay, only a tiny fraction of the potential inoculum produce land on susceptible host plant. That's it. There's nothing much to discuss on this. Moving on, penetration and invasion. After the attachment of an infectious propagule, we have discussed for virus, propagule means what? Inoculum. Okay, one unit of inoculum, it's called propagule. For fungi, it's spores, sclerotia. For others like bacteria, viruses, it's the whole pathogen body, right? So after the attachment of an infectious propagule, the penetration process may involve direct mechanical pressure exerted, for example, by fungal apresoria, enzymatic digestion of the plant cuticle, or both. Pathogens such as bacteria invade via natural opening. So natural opening, one of the example of natural opening is what? Stomata or any wound in the plant body. Direct penetration is most common. Remember this. Direct penetration is most common route for fungi, oomycetes, nematodes, and parasitic plant. It, it's not usually seen or not at all seen for in case of bacteria. They require open natural opening or any kind of wound for their entry. Okay. Okay. What is there next? Let's see one evidence what they are suggesting. Now, evidence suggests that many classically defined necrotrophs known to produce various toxins to kill their host undergo a brief cryptic biotrophic phase during which they initiate an intracellular hyphal invasion of their host, and host defense is being suppressed using various mechanisms. Here you will see one example, go through it. Sclerotium, Sclerotinia, sclerotium, they produce oxalic acid, and it is utilized to modulate hormonal defense and manipulate the photosynthetic machinery of the host to suppress cell death. Okay. What do we have next? The growth and reproduction of plant pathogens. Okay. Pathogens infect different parts of the plant based on their nutritional needs. Nutritional needs. Okay. Now, what they're saying, pathogens infecting above ground green tissues such as green leaves that act as assimilate producing tissues or sources employ different strategies to subdue their host for nutrition in contrast to pathogens that infect sink tissues to which assimilates are transported, such as flowers, developing fruits, or grains. Roots can be both source and sink for assimilates, and many roots and vascular pathogens are directly tapping into the flow of nutrient between above and below ground tissues via xylem and phloem. Now, this I believe this paragraph is a little bit used with technical words like this assimilate producing tissues, sink tissues. What are they? So, this assimilate producing tissues, the tissues which are normally responsible for photosynthesis, means they're responsible for preparing food for their plant. Now, the sink tissues, they are the food storing tissues. Okay. So the assimilate producing tissues, they prepare food, and the sink tissues, they stores food. In simple terms, if we try to explain this paragraph, is that some pathogens infect green parts like leaves, means the above ground green tissues, okay, which make food for plant, that is the source tissues, right? Other pathogens, uh, other pathogens infect plant parts like flowers, fruits, or grains, which do not produce food but store them. That is the sink tissues. Okay. Now, roots are special because they can act as both source and sink depending on the plant's need. Many roots and vascular pathogens means what? The pathogens which infect both xylem and phloem. Okay, this xylem and phloem are called vascular bundles, we know. Okay, so there are many roots and vascular pathogens that target the transport system of the plant. They tap into the flow of nutrients moving between upper, that is the leaves, and lower, the root parts. Okay, they target the upper part as well and also the lower parts. Simply, they have discussed it here. Okay. Okay, we can see one such example is that some pathogens classically defined as necrotropes. We have heard of this term necrotropes, which live on living, non-living, that is dead um, tissues. They use toxins, necrosis inducing proteins, or hydrolytic enzyme to obtain nutrition by causing cell death. Okay, they intentionally cause the cell to be death so that they can survive on that death part of the plant. So they release toxins, necrosis inducing proteins, or hydrolytic enzymes to obtain nutrition from by causing cell death of plant. Okay. Okay, here is it. Here it is. Effect of pathogen on photosynthesis. Now, photosynthesis happens in the chloroplast. Okay, we have studied it in 10 plus 2. But during stress, stress means what? It can be biotic stress, this term. We human have so many stress, right? Physical stress, mental stress, so many pressures. The same, accordingly, plants are having stress as well. It can be a biotic stress or biotic stress. Abiotic stress means the pressures that they feel due to the abiotic factors, the non-living factors, the temperature, humidity, rainfall, and all. Biotic stress means what? The due to pathogens, insects, and all those things. All right. So during this stress, chloroplast produce ROS. What is ROS? That is reactive oxygen species. Hmm? Now, very small amount of this ROS help signal the plant to start defending itself. Like if this is a plant and there is one pathogen, okay, pathogen, okay, I don't know what I tried to draw. Consider it a bacteria, okay, this is the flagella, bacteria. This is the leaf. So when a plant is under attack, a small amount of ROAs, they um, what they do, they help signal the plant to start defending itself. It's like a warning system, okay? But what happens? Small amount is fine, but what happens when it is too much? Like too much of this ROS can damage plant cells. So plants use antioxidants and photoprotectant to keep them in balance in a sense, so that yes, ROS is important because if they will not give signal, so plant will not understand when there is a pathogen and when to defend themselves. But anything too much is not really good for any of us, be it a pathogen, be it a plant or animals or humans, right? So too much of ROS is harmful. Again, they damage the plant cells. So what they use? They use antioxidant and photoprotectant to keep them in balance. Balance. Okay. Okay. Now, pathogens, like how pathogens use this against the plant. Some pathogen, what they do, they target chloroplast. Chloroplast is essential for this photosynthesis. The pathogen, what they do, they attack, they target the chloroplast. They target chloroplast to stop this ROS from building up so that plant cannot fight back. If they target chloroplast and this ROS it gets stopped, so they cannot give plant this signal. There will be no signal defend yourself, any foreign particle is entering, so there is no defense, and they are, helpless, and pathogen can attack easily. One thing that pathogen do against plant. Next, what happens, the other pathogen, the others increase ROS. There are one type of pathogen which stops the ROS activity so that they cannot send the signal. There are other type of pathogen. What they do, they increase the ROS, amount of ROS to kill plant cells. That is, if plant cell will be killed, then who will be benefited? If plant cells are killed, who will be benefited? So many times we have discussed. See, here only is there. If plant cell will be dead, necrotrophs will get benefited, and there are majority of majority of the plant pathogens are necrotrophic. So the pathogen, what they do, they increase the amount of ROS in plant cell so that the plant cell will be damaged and they will be dead, so that necrotrophic pathogen can stay and survive and absorb nutrients from that dead tissue. Okay. The pathogen, they send effector proteins to interfere with photosynthesis and hormone signaling. Understood? It's not that difficult. The main thing is photosynthesis is very important, and pathogen literally play with this photosynthesis thing. Either they decrease or they stop ROS so that they cannot give them signal, or else they increase it so that the tissue get damaged and necrotrophic pathogens, they get benefited. That's it. Next, there is same thing will be written here everywhere. Okay, just go through it, read it. The next we have effects of pathogen on photorespiration and respiration. First was photosynthesis. Now we will talk about this photorespiration and respiration. Now, photorespiration happens when a plant use oxygen. Plant use oxygen instead of carbon dioxide. Normally, if we know the use of the process of photosynthesis and respiration, we will know for respiration they take up oxygen instead of carbon dioxide, right? Especially during heat or time of stress. Stress means biotic or abiotic stress. So this helps protect the plant from damage by removing extra energy and harmful molecules. Harmful molecules means what again? ROS. Harmful means what? Excess amount of ROS will be removed. Now, pathogens, what they do? Pathogens often enter through stomata. We have discussed that again. They enter through stomata, natural opening, right? And cause stomatal closure. There are various role of stomata, right? If stomata will be closed, so one though which is coming to my mind, there will be very loss of transpiration. Water loss will be reduced. So let's see how they will be harmed. So what they will do, they will enter through stomata and cause stomatal damage, which will increase photorespiration. Photorespiration will get increased, and the production of H2O2. H2O2 is what? Now H2O2 is a signaling molecule for plant defense. So, this plant, they are very clever. They are not that complex, but they are very clever. Like, they have secured a boundary around them, and there are so many soldiers around it. Soldiers, ki anywhere pathogen is trying to attack, they have set so many signaling molecules, either ROS or H2O2, कि they give the plant signal कि enemy is coming, you start defending yourself. Okay, now pathogen, what they do, they target all those areas so that they cannot get those information, that message, so that plant will be defenseless and pathogen can attack to the plant. First part we have seen, they have targeted the ROS, chloroplast area. Now they have targeted the stomata so that photorespiration can increase and this H2O2 production will increase, which is a signaling molecule of plant defense. Now respiration. This is photorespiration done. Now what about respiration? Respiration is how plants break down sugars to get energy, right? Now during infection, what happens? This process speed up to meet the plant's extra energy needs for defense. In resistant plant, what happens? Whether it is resistant or susceptible plants, in case of pathogen attack, respiration increases. That is normal, okay? But there is slight difference between resistant plants and susceptible plants, okay? In resistant plant, what happens? Respiration rises too quickly and falls suddenly after that, okay? But in susceptible plants, what happens is stay high for longer period of time, okay? Respiration, I'm talking about respiration, okay? So in resistant plant, it picks up very high but then drops down like very quickly. But in susceptible plants, what happens? They rise slowly but they stays there. Rise up of respiration we are talking, but they stays uh, high for longer period of time, okay? Now, before some time, when we talked about stomata, we have discussed a little bit about transpiration also, right? Because transpiration is how plant lose water through stomata. They lose water through stomata while taking in carbon dioxide for photosynthesis. Yes, we know that. Now, when pathogen infect the plant, again and again we are saying ki what are the different physiological activities that gets disrupted because of this pathogen attack again. So when pathogen infect the plant, they often disturb the stomatal functions, blocking, opening or closing them. Means they play with the stomata. Now some bacteria and fungi use toxins or effectors to force stomata to open or close for their benefit. These are their weapons to force the stomata to open or to close. This obviously disrupts the normal water use efficiency, right? If stomata will be forcefully closed, so what will happen to transpiration? And if there will be no loss of water, then you can understand, right? Or if it will be open for longer period of time, it will make it lose more water and reduce carbon dioxide intake. Now, so what happened in resistant plants? Stomata may get locked shut to stop infection, but what happened then is stomata will be locked in resistant plants. This can, also slow down photosynthesis and reduce growth. So we can see so many plants which are stunted. So many resistant plants, they are obviously benefited because they are beneficial because many pathogens cannot attack there, but there are so many side effects also, right? So that resistant varieties can be stunted. In general, what we can say, disease reduce transpiration efficiency, especially in severe or long lasting infections. Okay. Let's see how pathogen affects translocation here. Now, some pathogens damage or block xylem vessels, which carry what? Xylem is what? If this is a plant, above ground, below ground is roots, and here we have xylem vessel, which absorb water from roots and make it available to the plants. Water and mineral, they absorb water and mineral, leading now if it will be blocked, some pathogen what they do, they block this xylem vessel, so there will be no intake of water, right? So if there will be no intake of water, that disease or that symptom can be called as wilting. Wilting is mostly caused by soil-borne pathogens. The one of the major pathogen which is responsible for wilting is Fusarium wilt. But wilting can be caused by various factors like fungal wilt, bacterial wilt, physiological wilt. Okay, so we cannot say so sure before testing it under laboratory conditions. Now, what plant can do to defend them? They can defend by forming, where should I write? Tylosis. This is their defense strategy against pathogen who block the xylem vessel. So plants defend by forming this tylosis, which in normal sense we can call as xylem plug. Okay, or gums or reinforcing tissues with lignin suberin to stop the spread of that pathogen. Now, pathogen also interfere with the phloem. Two vascular bundles are there. One is xylem, another will be phloem. So they also interfere with the phloem, which carries sugars, foods, right? So some pathogen turns leaf into sink tissues, means they convert the source tissues, means the food preparing tissues, to sink tissues, means the tissues which are responsible for storing the food. What the pathogen do? They steal. They steal sugars, weakening the plant and lowering defense. Now, two things they do. One is, so they are stopping the xylem vessel so that they cannot take water and mineral from the root. The second, what they do, they convert the source, convert how they use toxins or effector proteins, so they convert this source to sink tissues, and they steal the food. They steal food so that the plants get weakened and they are not ready to defend the coming pathogens. These are all the strategies, the war against pathogen, versus plant. Huh? So they are using their all strategies, all the weapons, uh, toxins, effectors, and all the thing and strategies to make the plant weak, whereas plant is using all the different strategies, H2O2, ROS, so many signals, so that they get ready for when and only the pathogen will come and attack, they can defend themselves. So let's see, it will go on. Who will win? We have no idea. But how they play this game, how they strategize this war, we are studying it. So try to remember it in like a story, not like study. What pathogen is doing, how they are fighting a battle, and how plant is defending them. Try to remember it like a story, like COC. We know games, right? Clash of Clans. Okay. Hmm, next. Again, effects of pathogen in on what? Plant growth and reproduction. Now, plants disrupt plant growth and reproduction by altering hormones, okay, damaging tissues, redirecting nutrients, and destroying reproductive organs. This leads to what? This leads to dwarfing of plants, means they are stunted. Hmm? Poor flowering, fruit loss, and reduced seed quality. Ultimately, what happens? It lowers the yield and survival. Now, let's see one example. This is one of the example, okay? Because this agrobacterium, A is for agrobacterium. Agrobacterium tumefaciens, this pathogen is responsible for crown gall disease. In roots, you have seen, right? Like tumor-like appearance, like this balls. Hmm? So it inserts its DNA. The pathogen inserts its DNA into the plant, causing these tumor-like projections. Huh? These galls. By what? By interfering with hormones. Now, what are the plant hormones that we know? Plant hormones are auxins, gibberellins, cytokinins, right? So what they do? They interfere with auxins and cytokinins. Auxins, why only auxins and cytokinins and not the other hormones? Because auxins and cytokinins, they are responsible, cytokinins, they are responsible for the root growth. So they target these auxins and cytokinins, which diverts nutrients and slows growth and damage tissues. Simple. Next, one more example can be of fusarium. Hmm? Fusarium species, they normally cause wilting or seed blight or head blight. Normally, it is responsible for wilting. Okay. So there are many other examples that you can search and study, or if you want to know, you can just ask me in comment sections, or any more clarification or more understanding or any point that you are missing, or any doubt, just comment down. Overall, what we can see here, the infections reduce plant size, vigor, reproductive success, which directly impacts agricultural productivity. Everything that is happening here, ultimately it will hamper the agricultural productivity. Okay. Now, how they affect the biomass development and yield. Pathogens reduce plants' overall growth or biomass by what? By damaging leaves. If the leaves will be damaged, overall biomass will be reduced. So by damaging leaves, what they do? It lowers the light absorption. If there will be less number of leaves, so there will be less absorption of light, which will hamper the photosynthesis. Hmm. This is about above ground. Below ground, what will happen? They infect roots, which limits the uptake of water and minerals because they are targeting xylem. Again, mm, if there is no absorption of water or mineral, and we have also heard they target phloem as well. If they target phloem also, and they steal the food, they convert the assimilate tissue to sink tissue, so there is no availability of food and plants get weak. again hampering the overall development and yield of uh, a crop. With example, we can say, um, let's see eye spot disease in wheat. Anybody heard of it? Eye spot disease? This is not a very common disease of wheat. Normally in wheat we have so many major diseases. Okay, it is normally it is caused by oculimacula yellow day. Okay, it infects the base of wheat stem, which weakens the stem after flowering, causing lodging. Lodging means what? If the plants, falls like this, the plant should be straight, right? So plants fall over. The term is called lodging. It's the agronomical term. It interrupts nutrient flow of the grain, leads to whiteheads. Whiteheads, how it will look like the symptoms, like empty or poorly filled ears. Final result, how it will, what it will happen? It will lower the grain weight and again yield loss. See how a single plant can affect so many physiological parameters. They target everywhere from top to bottom. In this picture you can see the growth, see the galls. Hmm? Study the images. I have told you, study images. Images is everything. From there so many questions comes. Okay, so this is the end of chapter 2. We have discussed the effects of pathogen on photosynthesis, respiration, transpiration, translocation, reproduction, and total biomass. I have added the takeaway questions in the description box. Make sure to check them out. And if anything still feels unclear, just drop your doubts in the comments. I will be happy to help. Thank you so much for watching. If you found this helpful, please like, share, and subscribe. And don't forget to hit the bell icon to stay connected with our PhytoPathoVerse family. See you in the next episode. Till then, keep learning, keep growing. See ya.